for biotherapeutics with expanding responsibility, like quality assurance, quality control, manufacturing, supply chain, key account management of device partner companies, global medical device surveillance. Present position, global device project leader and drug device integrator. His special interest, pioneering a new field in biotherapeutics, such as drug device integration during research and development phase, gaining new insights and learning in biotherapeutics and drug delivery device technologies, building and forming new concepts, processes, and establishing cross-functional diverse teams. Welcome.
Thank you very much. Also, good morning, good afternoon already. It's a real pleasure for being here, and first time with Scholar conferences, the first time for me being outside the European bubble. So after the pandemic, as for many, it has been difficult to travel. And well, the first action I took when I was arriving here was climbing the Burj Khalifa, which is, you know, in terms of innovation, quite impressive. So it's really something you can be very proud of. I would like to provide you some sort of overview on drug-device combinations. We talked a lot about drug delivery drug combinations, and now I would like to add one more dimension to it. And I would like to first provide you some sort of framework, some innovation of what is available here, and then focus on two aspects. The one is the formulation, the drug formulation, and then also the kind of medical devices. However, I want to keep the protein formulation part relatively short, because in the end of the day, it is the combined product that you hold in your hand, and the face, so to say, of the product is really then in the medical device part, and that is the part where all these entities flow together. So I would like to make that my emphasis for this speech here. The starting point for the significance of drug-device combinations has been really the advent of the biologics as becoming a more set of drug entities in the pipelines of the drug manufacturers. Why so? Because biologics are more efficacious, they are generally safer, and they also provide you access to new kind quality of therapies. And they are, in that regard, advanced to the conventional chemical drug that you have on the market in a more targeted manner. But the downside of it is that they often require a parenteral route of administration. And so you typically then result in a high demand on injectable drugs that you have to take care of. I would like to focus here, because it has been become popular, you know, to introduce the monoclonal antibodies as a sub-part of those biologics. And as you can see from the number of entities out there, you know, when new approvals, they are clearly increasing here. And that trend will continue as it stretches into new therapeutic areas. Now, what is the scene here we are talking about? You have, of course, first of all, the patient in the center, and then you have the drug formulation, and then finally the device. This, you know, simple picture implies quite a few things, which gives you a kind of projection in terms of the clinical requirements for each of those arenas, and also from for the development and the regulatory part associated with it. If you look at the interactions between the device and the patient, it's in the center, the ease of use. So you want to have an adherence to the therapy, you want to make it to be used safely and efficiently. Then between the device and the formulation, you have a kind of design space. I will come to that in more detail. And of course, then you have between the formulation and the patient, the physiological aspects, of course, and also the physical-chemical properties of the formulation. The challenges of those protein formulations are coming along with the chronic diseases that are increasingly applying medications that are being administered by the patients themselves. So a chronic disease where you need to maybe sometimes lifelong therapy, you then have to think about self-administration to make it, as much as possible safe and kind of user-friendly. And the delivery of those doses, I mentioned protein formulations are often provided parenterally. You need to use prefilled syringes or devices like pens or auto-injectors. Now, if you look into monoclonal antibodies, what can be seen here is that the dose is really increasing here and challenging the fill size of the pre-filled syringes, which typically are 1 milliliter, what you typically have. And so here you have, from the formulation perspective, asking from a clinical side for larger doses, you really have a challenge here. And then you have, on the other hand, also the challenge that the administration of that liquid is quite limited in terms of volumes. So, and in addition, you also want to be sure that the bolus is given typically fast, but sometimes you might also require an adapted ejection rate if you think about a sustained release, for example. So combination of high doses and limited volumes, you then have this mentioned challenge of drug concentrations for monoclonal antibodies that range above 100 and even up to 200 mg per ml, which typically is also then defining the upper limit here. What you then see still is that you have aggregation. That is what likely might occur if you go to these borders of the concentration that you can generate, as well as you then have, in many, many cases, increased viscosity. Now, let's look at the first step in trying to achieve a stable protein formulation. That is, something that is where you can then dissect it into subgroups, like the chemical instability. That is an area where you have to be aware of, like oxidation, hydrolysis, deamidation, and so on. And then you have physical instability aspects like conformational changes or colloidal activities that might also endanger the protein stability. So looking at the complexity and the stability that wants to be achieved, you really have to look into the kind of behavioral aspects in terms of the chemical and physical-chemical aspects of the drug compounds. This is then what you usually end up with when in the development of antibody formulations. That you start with some sort of proto-formulations where you look at the various buffers isotonants to get all the physical chemical aspects right. And then you continue by further looking into the high throughput evaluation of those proto formulations and try to rank the best of those candidates that you have for formulation candidates in the end that will finally make it to a marketable drug formulation. And then the $100 million question really is, when would be the right time for the decision of thinking about the administration of this compound? And that is then right here, whenever you start with accelerated stability studies of your protein formulation and a selected primary packaging. That is really the point in time when you then also have to think about how will my formulation then in the end also perform in terms of a pen injector or an auto injector or a kind of safety syringe or anything else of a device that you might think of. So here is exactly the point where a device team would have to start co-working with the formulation team. And then if you do that properly, that you then have already some sort of an integrated understanding of the primary packaging. Because once the product is in the primary packaging, so whenever it's in the pre-filled syringe, changing afterwards is doable, of course, but it means that you have to typically start all over again. And this is a nightmare, and you lose time. It's a lot of money and typically not found to be acceptable. And if you are in the commercial arena, you then are confronted with, well, your drug fails simply because at the time we will come with this drug to the market, well, somebody else will be there already. So I mentioned that the viscosity is really something of these formulations in the end that are quite challenging and to be aware of. Also, when thinking about putting this formulation ready to administer via a syringe or other kind of device. So you need to very much understand the viscosity, which of course is highly dependent on the MAP concentration. And what you should also think here is about that it's not a linear function, it's going up exponentially. This has decisive consequences in terms that if you look here, 150 mg per mil, it's comparably low, so 10 centipoise is something that you would still see as quite acceptable. But then, you know, between 150 and 200, it already, the curve incline already starts to become quite challenging. Plus the fact that these biologics have to be stored in a fridge. So you have in a cool chain. And when you take those out of the cool chain, well, then you also have typically higher viscosity to cope with. So that on top has to be considered here as well. So what can you do then in order to reduce that viscosity? That is really an art for itself. You have to think about really how you can best combine the excipients that you use. And there is various means to do it by changing the salt concentration in the product without, of course, compromising on the physiological requirements of the formulation. So, and you typically then end up with a kind of experimental design. And I have from practical experience, I have to say, you also often work even in industry. You work with specialized companies here who really have enough overview on the available excipients, because it's not just the excipients as such that should work from chemical or physical chemical properties, but you also have already think ahead about the regulatory acceptance. And you might not have, as you mentioned this morning, all the data and know-how available in your own organization. Sure, you would probably want to reach out to somebody else who might have that, and at least can give you some sort of hint in which direction to go. So the viscosity control, very important. And then you really need to also think of the primary container here in terms of the device. So what you should do is, or what the formulation departments typically do, is they conduct some experiments in what they call the syringeability. They look how much force it takes, you know, to squeeze out all the contents. But at that point in time, you should also bring the device team with you, and they should do some additional experiments with it in a more way that takes into consideration all the user-related aspects. So in the end, you should then start all, and you should also work with your primary container supplier here sometimes, because if you have to, for let's say, auto injector, because somebody is not able to press, you know, the 10, 12, or 15 Newton to administer safely with a syringe, well, you might think that an auto injector might be appropriate. And then you really need to design it in the way that it does not compromise on the stability, I mean, the glass actually of the primary container, that it doesn't break, or that you have some sort of integrity issues that, you know, put at risk the sterility of the product, so micro cracks, for example. And last but not least, you should also think about, well, you concentrate as much as possible. Because you want to shrink the volume, and even then the requirement might exceed not only the 1 ml or a 2 ml pre-filled syringe, but go even higher. And then you really have to think about other devices than pre-filled syringe-based devices like pens or auto-injectors. And that is so because you will then have to look at those special needs of typical therapies that have come into scope. We talked about this morning already about rarer diseases. Hypercholesterolemia is one of them, but some subsets are really rare, considered to be rare diseases here. Multiple sclerosis, rheumatoid arthritis, SLE, and IPF, and there are many more coming up in the pipelines where you can get a more granularity on the disease backgrounds, and that might also need adjustments in terms of the dosing volumes, which then call for certain suitable devices to administer. Now, if you go beyond that, what is literature is typically described as 1 ml or 2 ml at the most. You know, what is out there? So what is really large volume? If you go out and look, you then have these kind of drugs that you find on the market already, which go for concentrations up to 50 or 200 mg per ml. That comes still in pre-filled syringes. But you then have at the other end of the spectrum protein formulations that come in concentrations also at the same rate of 200 mg per ml, but in much larger volume. So 50, 100, or even more in total volume to be administered. And do you need to do then? You can then, of course, change the primary container, make it bigger, and have some sort of different mechanism behind it in order to administer. And or you can also think about enabling technologies by adding viscosity reducers like hyaluronidase, as Herceptin, that would kind of require a reformulation. So as I just mentioned, thinking about the volume, so regular handheld syringes, 1 milliliter, maybe a bit more than that. Then typically pen systems, auto injectors, also quite convenient, 1 up to 2 milliliter. But then you have these kind of infusion pumps that you find there in the market that are already available. But in between this 2 or 3 milliliters and up to then typically 10, 20 or even more milliliter, you still have some sort of a gap here. And comparably, quite a number of products and drug candidates fall into that category. And if you look into that, you then see some of those I have put on here. You typically cannot operate them manually then anymore by holding and pressing them down for the time of administration, but you have to put on the skin and then press a button for a longer period of time. So kind of patch pump based systems that you then have to typically think about, and which are more and more now entering the market from various suppliers. Another aspect I have not really given much of focus up to now is the pain challenge that's associated with it. And the pain question is something very complicated. It is partially, of course, based on physiological aspects, like the needle gauge or the injection volume and the speed of the injection, besides the drug formulation and its physicochemical properties. But it's also, of course, a kind of psychological, so it's in the emotional aspects of thinking about needle phobia of patients, just the understanding or having in mind a needle is entering my body is causing some sort of fear. There has been quite some work done on that aspect. One of the most fundamental is the work by Jorgensen. But you know, you can use that as a basis. It has more or less been in focus at the time, still up to 1 milliliter. If you now think beyond 1 or 2 milliliters, you really have to do that in kind of a clinical setting for your drug and for your device in combination. So that is really something that you need to intensely study for the specific therapeutic targets. So it's not that you can say, well, I do it just once, and then for any other therapeutic area I have in mind, it will be just the same. So you really have to look at the therapeutic target. You really have to see at the drug candidate, and then you have to work consequently your way towards it to assess it properly from a clinical perspective. Now, I mentioned that at the point in time when you have your highly favorable candidate of drug formulation in the chosen primary container, that is then really the time also to think about the device. What does the device team, in parallel to all the clinical work and the formulation work that is still ongoing, do? They kick off a kind of a process that's based on engineering requirements. What you see here is a kind of waterfall, a diagram that you see also by the regulators like the FDA. They have that in their guidance, you know, how to design the right device for the target indications. And that process typically starts with a kind of pre-project before you then enter into a kind of design control, so a very regulated process, which then is going to become part also for the submission, the packaging for the NDA in the end. So like for the drug formulation, where you look for proof of principle of the drug candidate in the clinical setting, you do all that on the device side. You then, it's a process that typically takes about a year, even for those systems that are available already for other drug candidates. And then it takes another year or 2 years that you then come to a proven proof of concept stage. Before you then, with the clinical data, proof of concept clinically, that you can then, well, everything is fine. We have the proof of concept, we have the right drug, we have the right device, and now we go into full commercial scale here. So that's where you put the big money on the table and say, let's go and let's bring the product to the market. So that's the kind of showing how the process works, that you look at the various angles in what is already out there. You really have to watch also the competition very carefully here. The market needs have to be examined, of course, but also alongside with it, especially if you are in the commercial arena here, you have to look at the competition and see what is their concept they are going to position and place on the market. If, for example, it is the same kind of profile in terms of clinical results, well, they might have it much easier to administer, maybe just once a month, and you come with a weekly or a daily even administration, well, you might not likely be that successful anymore. But these questions are sometimes very tricky and challenging to answer. So here also, besides clinical studies, you also need to think about a lot of market research and meta studies that you have to conduct. Okay, what you do then is you come up with a set of criteria what should be fulfilled. So-called user requirements. So what is really required to make this product a success from the perspective of the patient, of all the healthcare professionals alongside that might also have vested interests here in terms of reimbursement. So the pricing here is something really the user and their needs. And you really have to thoroughly look into that, and you have to list those and convert those into a kind of technical specification in the end. So what kind of features should your device then have? And you can do that in a kind of scorecard framework where you rank and grade all these criteria. Then in technical terms, you need to quantify that. And then once you have undergone this, you will have your favorite system available. So what you do then is really, you look at the formulation first, then you come up with, whenever you have the primary container fixed with the device part, and then you push it on further up until the clinical proof of concept, which is typically then also the point of decision, full development, yes or no. I would like to also highlight that this approach is, even for big pharma companies, quite an integrated approach here. And it's also a challenging approach because you have to merge the formulation process requirements with the requirements and the related processes of a medical device. So both kind of processes sometimes really have to be integrated. And typically you do not have this technology in-house, especially when it comes to devices. It's sometimes, and I would say the majority of even the top pharmaceutical companies work here with device partners. Those have to be tied in very closely, and you have to collaborate here for this one drug product, what it is going to be in the end. Yeah, you then have, whenever you have the kind of understanding on the formulation side and also on the pure device side with some dummy formulation, you really have to go then and do some integrated work here. You see that you then have prototypes available, and you see, because the formulation at this stage, you do not have enough in quantity, besides the fact that it is extremely expensive, but you then have not more than maybe 50 or at most 100 of those devices available. So those test designs have to be carefully assessed and worked out very properly to then come up with hopefully positive results to move on. Yeah, and throughout that cycle, it is also important to not to forget on that journey the patient here in focus. So what you do is you have to look at the human factors engineering very thoroughly. On the other hand, you have to look at the risk associated with it, because typically what is very easy and straightforward from a handling perspective is, from a risk management, maybe a showstopper. So you really have to bring those two together and integrate them in a kind of Deming cycle here. So you have to use prototypes, you have to test those with the patients, and then whenever you see there's shortcomings, you have to evaluate and assess them very carefully and see what it means in terms of further improving, optimizing your concept, so that at the end of the day, you come up here with a safe and patient-friendly device. Now, outlook. What is the essential part of it, and what to watch out for the future? First of all, the drug-device combinations are getting increasingly complex. I mentioned here with an example the monoclonal antibody formulations in safety syringe or in a pen or auto-injector, or maybe even a large volume device, kind of patch pump system. But the next level of complexity technically is already approaching, which is connected devices. So what you typically do, because these medications are extremely important, if you think about a shot costs you 10, 20, 30,000 euro or US dollars, you really want to make sure also from a reimbursement perspective and clinical perspective that this has been used successfully and effectively. So you want to have some reassurance, and that you can do with connected devices. So you transmit the data from the patient and the device to the doctor, for example, but also for monitoring. Then stakeholder management is very important in that context. You typically work together with a device partner, and it's an external company. You need to secure that very safely in terms of what does it mean in terms of IP, what does it mean in terms of manufacturing, etc. And that you have a smooth cooperation throughout the rollout of the product and thereafter, when you hopefully will get increased sales figures. And last but not least, really your organization, you have to think about effective and efficient structures here. Typically what you see, that drug product manufacturers have their own drug focused processes, and then you bring in the device, typically with a partner that has an own process. And the best really is here to integrate the two. You really, and because you are the contract giver here, because in the end of the day, your product that you bring to the market, you have to make sure also in terms of facing the regulatory bodies that you have to full control under this integrated process in the end. And it's of course also a matter of efficiency in the process, because you always want to go to the market in shorter periods of time and of course more cost efficient. So you have to start early, you have to think about the concept first to make it a success. Having said this, I want to close, and I'm of course ready for some questions from you.
For this informative presentation.
For this informative presentation, especially for me myself, I always work in small molecules, so it's interesting to see the other part of large molecules. I'm just wondering regarding the viscosity of the cells that are active. I'm wondering, is there any pipeline or specific limit that usually you try to target when developing the formulation that not exceeding that in terms of viscosity?
You mean the viscosity, right?
Yes.
Yeah. So what you can typically see is that if you go beyond 175 mg per mil, it's simply from the practical experience, it's usually extremely rare. 150, I would say, is feasible. 170 and beyond 200, I would say that is already the exception. Because the injection force is, you have stability challenges, but let's assume those can be controlled. You then have high viscosities, especially when it comes also, if you take into consideration the refrigeration. You have to put them into a school chain, and if you take them out, and even under normal condition, it's still workable. You are already in this incline of the curve here, and that really makes it almost impossible to go beyond that.
So the Q100 is the maximum.
Yeah, and the viscosity, I would say what can be handled is about 10 to 15 centipoise. Manually, depending on the patient population. If you have elderly patients in many of these diseases, you have elderly patients which have some shortcomings. Well, you probably are then better off with kind of mechanically driven administration devices.
Thank you very much.